Welcome back. Let's finish part 2 here. We're going to go ahead and look at the sequence of the transcription in prokaryotes. We're going to compare eukaryotic and prokaryotic transcription. Some of this I think you do not know, whereas you might have known all the stuff we talked about before. I think some of this might be new, so please ask questions if you need them when we get back to class after spring break. Transcription synthesizes RNA using a DNA template. This was the definition we gave a week ago. DNA is mostly for eukaryotes in the cell's nucleus. The ribosomes are not in the nucleus. So we had this problem where we knew that the information from the DNA was converted into something that caused a polypeptide. It was converted to amino acids. But there has to be an interim molecule in between, in eukaryotes at least, that takes the information from the inside the nucleus and transfers it to the ribosome. RNA in eukaryotes is synthesized in the nucleus as mRNA. This is a message that travels from the nucleus to a ribosome. In bacteria, we don't have a nucleus. And so we actually don't need to do all of the things to the mRNA that causes it to be able to pass through the nuclear envelope, but we still need that message. So bacteria still make a DNA, open up the DNA and make an mRNA transcript. That transcript is sometimes simultaneously translated by a ribosome. They're both in the same location. For eukaryotes, that is not an option. The enzyme that does this in eukaryotes and prokaryotes is called an RNA polymerase. Polymer is a strand of monomers. Ase is an enzyme. RNA is what this polymerase builds. In bacteria, we really only have one RNA polymerase, and this is what you learned in previous classes, I am sure. Its job is to synthesize mRNA. It does not need all of the other helpers that we need it during DNA replication. We're going to be able to grab onto the DNA with RNA polymerase, open it up, and just start elongating. Why don't we need a primer? The primer that was required in DNA synthesis was an RNA primer. It was made out of primase. It was made by primase. We needed it because one of the rules of DNA is that DNA can't just be attached with hydrogen bonds. It needs to form a phosphodiester bond with a previous free 3 of a nucleotide. RNA doesn't have that rule. RNA was our primer in DNA replication. So now if I'm building an RNA strand, all I need is to add the RNA. The RNA polymerase that we use in bacterial transcription it has a core enzyme here, and the core enzyme is made of an alpha subunit, beta prime, and an omega unit. I actually want to highlight a few of these later, so I introduce them. But the most important thing to you is this sigma subunit here. This is a sigma, this letter. This sigma subunit is what is actually doing most of the attachment and catalytic activity that we're going to talk about. These little NTPs are nucleotide triphosphates that are going to be RNA nucleotide triphosphates. It's going to build a complementary anti-parallel strand of RNA that can then leave the nucleus and head out into the cytoplasm if you're a eukaryote, or just head to the ribosome if you're a bacteria. The transcription start site is on the DNA. It is where we want to start making a copy so that I can read the gene and bring the code to the ribosome. The first thing we need to do is denature the DNA itself, and we want to denature it by breaking those hydrogen bonds between the adenine and the thymine and the cytosine and the guanine. This makes that area accessible for RNA polymerase. So RNA polymerase can read the template or the side of the DNA that is actually conveying the information and make an mRNA transcript or an mRNA copy of it. The promoter region is on the DNA. It is a region basically marking where the gene is about to start. It's upstream of the gene, so it means that when the polymerase grabs, it's going to be able to move in the correct direction and sequence the gene in the correct order. When the polymerase with its sigma unit attaches to the promoter region, this can be a little bit wobbly, or it can be a little bit stronger held on. The efficiency of its ability to hold on and clasp onto this promoter is actually, it's a feature, not a bug. When you guys talked about the lac operon in micro, one of the ways that we regulated the lac operon in bacteria, and you did negative and positive regulation. Positive regulation was asking how many times do I actually transcribe this transcript. That was actually regulated by how good the RNA polymerase was at attaching and holding on while it was actually making the transcript. The more the cell needed to make those transcripts, the more things the cell did to make the RNA polymerase hold on tight. If the cell kind of needed to make the transcript, it didn't do a lot to facilitate the transcription process. If it really needed the transcript, like lactose was the only food available, then the cell would do a lot of other things to regulate and strengthen the bond between RNA polymerase and the promoter and the subsequent template strand. The promoter region is actually recognized by this sigma subunit so that they bind together. And then if you look here, as RNA polymerase is activated, it opens up the strand of DNA and begins the process of transcription. Promoters are sequences of code that play a role in telling RNA to attach to that particular area because there's a gene downstream. Promoters are often consensus sequences. That means it's a sequence that is the same everywhere it's a promoter. Consensus sequences are common in bacteria, less so in eukaryotes. They are areas where I have a long strand of the same A's and T's and C's and G's, often ATT, ATT, or longer sequence of T's and A's than C's and G's. That's a region that the promoter or that the RNA polymerase recognizes as a promoter spot. Let me say it a different way just in case. Consensus sequences are sequences that are the same but found in multiple places on the genome of a bacteria. They mean the same thing even though they're in different locations. They all mean this is a promoter in this case. The Pribnow box, TAT, AAT, is a really common sequence in really recognizable locations, which tells the protein involved, the sigma subunit of the RNA polymerase, to attach there and start transcription. If you like this lecture is very vocab heavy, please ask me if this gets confusing. We've defined all these words and we've talked about them, but I feel like I can hear it when I'm talking. I feel like almost every word out of my mouth is now a vocab word that we've previously had. So please, we could absolutely go over this in lab again. Please ask me if this gets confusing. I think this is where we really get into the meat of all of the stuff we have learned. After we attach the RNA polymerase to the promoter region of the DNA is opened up. The very first RNA nucleotide is placed, and then the process of elongation occurs. Elongation occurs in the 5-3 direction, so we still have this rule that we can build on the free 3 side only, because that's a nucleotide rule. We build on the 3' side of the growing RNA transcript sigma will fall off. So this little attachment protein that was part of the RNA polymerase falls off. And then RNA polymerase does its thing. It adds about 50 RNA nucleotides per second, an A, a C, a G, or a U, by reading the DNA of the template side and matching it complement to complement. It also does some fact checking, or some, not fact checking, error checking on the way. It does some proofreading as it adds. If it realizes it makes a mistake, if the molecule that it is creating is not holding on completely solidly with good hydrogen bonds, it'll actually backtrack, excise out the wrong nucleotide, and place the right nucleotide in. So it acts as a proofreading and elongation molecule at this point. It keeps going until it encounters the end sequence, which is a termination sequence. In bacteria, that termination sequence is actually transcribed. It continues to copy until it continues to transcribe until it gets to the very end. There are two ways this process ends in bacteria. One of which is the sequence that it reads as it gets to the end of where the gene actually is important. It gets to a series of C's and G's. And so as the transcript is being made, because it's making a G, G, C, C, G, G, C, C, maybe of itself, as this starts to be spit out of the RNA polymerase molecule, these guys kind of fold around and bind to each other. So it would form, see if I can draw it, G, G, C, C, and then it goes around and it will form G, G, C, C, and wrap around on itself. When it does that, it forms this little hairpin turn, or a loop-de-loop. I had always learned it as a hairpin because it looked like a pin of hair stuck through the molecule itself. This fold messes up the speed and the rate that RNA polymerase is doing its thing, and it causes the RNA polymerase to fall off. This is an intrinsic termination. It's built into the molecule itself, the building mRNA sequence, that this is going to be a termination sequence, and it's based on the literal structure of the molecule. We're forming hydrogen bonds between members of the mRNA that forms a loop that actually interferes with the process continuing, so the process stops. In other situations, we have what's called a rho-dependent termination. Rho, this is the letter rho, it's a 6-part protein. The word for that, I don't know if I said it before. I want to say it now. It's a protein made out of 6 different polypeptides, or 6 of the same polypeptides, called a hexameric. It surrounds the RNA that is being made. It binds to a specific code on the newly transcribed mRNA called a Rho utilization site, or a RUT. I don't know why it's not a RUS, but it's a RUT. And when it binds, it actually starts chasing it as fast as it can and sliding up the molecule. It even makes that sound. And as it does it, as it runs into a hairpin turn, it actually cleaves the molecule so that it breaks it off. Not maybe 20%, I did say 20% of the genes, the mRNA that are being transcribed, actually involve a Rho protein. The other 80%, I'm going to bop back, use just this intrinsic termination where the shape of the newly transcribed mRNA interferes with the process. It's kind of cool, though. Versus bacteria versus eukaryotic transcription. Obviously, eukaryotes have a whole level of complexity that bacteria don't. We've got way more chromosomes that have information on them. We have our chromosomes in a nucleus protected by a phospholipid bilayer, so we can't just move things back and forth between the ribosome and the DNA. There's a literal barrier in the way. The DNA structure of a eukaryote involves a lot more wrapped up tight proteins twisted around the DNA molecule itself. It's often stored as chromatin, even if it is euchromatin, which we talked about a week ago. That euchromatin is still wrapped around histone proteins in nucleosomes. That means we have to be able to get at it, and get at it in a way that we can access all of the nucleotide sequences in our template strand, the strand we want to read of the DNA. RNA polymerase actually relies on a whole bunch of different transcription factors in eukaryotes. I'm going to give you some highlights of them, but things that help transcription proceed or don't allow transcription proceed. We don't have things like operons. We're not turning things on and off with a switch. And as we kind of mentioned the other day, that's awesome in a multicellular organism, because if I get dumped in a vat of milk, I don't want all my cells starting to make lactose metabolism proteins. I want to regulate the process of digestion to a very small part of my body. That means operons are not going to work for me. I need to have a way to tell the cells in my intestines to do something different than the cells in my skin. And so those are going to be things like silencers and enhancers and transcription factors that not only turn genes on and off, but turn genes on and off at different times of your life and different parts of your body in different ways. We have 3 forms of RNA polymerase that we find consistently in eukaryotes. RNA polymerase 1 makes ribosomes. It synthesizes the rRNA. It happens in the nucleolus. That, if you remember, your nucleus has, in your cell, your nucleus has your DNA, has chromatin mostly, and then it has this kind of little dark spot that's a nucleolus. The nucleolus looks different under a microscope and when you stain it, because it has a different type of molecule in it, and different molecules stain slightly different. The nucleus contains a lot of RNA, including the ribosomal RNA that is going to be assembled to make ribosomes. And RNA polymerase 1 manages that. RNA polymerase 3 makes short-stranded RNA and tRNA. I'm not really going to talk about this guy, but I am going to talk about this guy. RNA polymerase 2, also called RNAP2, makes mRNA and SNPs of RNA as well. I'm going to mainly talk about this a little bit. The efficiency of RNA polymerase 2, RNAP2, is determined by 3 things. I just want to mention a few of them. We're going to highlight some as we go along later in the course. I also want to point out that these are very specific to the types of genes you are making. So as you study other processes and physiology and things, you may talk about things like silencers or enhancers and the specific ones that control the pathway that you're focusing on at that moment in the class. We have proximal promoter elements. Those are proximal means near to the center, near the promoter elements that are actually controlling the efficiency of the RNA polymerase, whether it can bind at all, and whether if it's bound, can it actually start. We have enhancers. Things like testosterone is often an enhancer. It has to bind a certain number of testosterone to the beginning of certain genes in order to make that gene even accessible for RNA polymerase to work. And we also have silencers, because one of the mechanisms we have is if you do not need to make the gene, you can actually put molecules in the way, maybe even far upstream from your gene, or you can do things like make your gene less polar, less accessible to mRNA transcription to even happen. Those things can silence or stop the production of that gene or slow it down. Enhancers increase transcription. Silencers slow or stop transcription of that particular gene. The core of the promoter region for most eukaryotes is going to be something called a Tata box. This is a region that is heavily T's and A's. This is thymine, adenine, thymine, adenine. It is on the DNA. as is all of this. All of this is DNA structure that is determining if we are going to transcribe and where to start. I just wrote this here and I realized there's another picture that's going to appear. Here. Oh, it's good. It's over here. I pulled this from a different textbook, but your textbook doesn't actually show all of these in action. I changed this from its name. The textbook that I had these actually named, so I pulled their names out, which is why this looks different. If my gene is actually here, I have a promoter region that includes a Tata box where my RNA polymerase 2 is going to attach. Upstream, before I'm actually getting to this promoter region, I might have all of these far away control elements. They're distal control elements, and they may include regions of the DNA that bind to specific molecules like hormones or other things that your body is making that then will ultimately require the production of that gene's polypeptide. As they fill up, this region, when it gets its third, in this picture, enhancer, it will actually be folded by a DNA bending protein that'll snap this to the promoter region. There's a series of enhancers or silencers that can play a role here. We're calling them like general transcriptional control proteins. When this enhancer binds to the RNA polymerase and the transcription factors, then transcription can start. So when you are controlling the transcription of your genes, you're not looking for a switch like an operator. You have a whole series of things that need to occur in some cases in order to allow that gene to actually transcribe. And so you can control it with a little bit more accuracy and subtlety than something like a bacteria can. Once you are done, and frankly, the next part of transcription is very similar to what you did in a bacteria. Your RNA polymerase 2 reads the DNA template. It makes a transcript from it by matching A to U and T to A and so on all the way down, building a piece of mRNA until it reaches the end. At the end process, and it depends on your gene, at the end of that process, you need to prep it to leave the nucleus and to go through its next couple steps before it is actually read by the ribosome. One of the ways you do that, you start out by adding a cap. You add a cap that's actually a guanine cap. It's 7 methylated guanines. You add them to the 5' side of your mRNA. That cap protects the transcript inside of your nucleus. Otherwise, your nucleus will break it down. It'll see it as an errant molecule. Let that ambulance go by. It absolutely allows the mRNA transcript through the nuclear pores. The nuclear pores are the pathway out of the nucleus, but unless you actually have a cap, the pathway will not open for you. And when you get to the ribosome, the cap allows the binding of the mRNA in the right location on the ribosome for actual reading of it. You then add a tail. Your tail is added by an enzyme called poly-A polymerase. It's going to add in sequences of 50 to 150 adenines at the end of your every single transcript, except for histones. They don't seem to get these. But everybody else seems to get these polyadenated tails. As far as we know now, these polyadenated tails are added in chunks. You have 2 or 3 or 10 steps of adding these polyadenated tails. It is one of the ways that you can regulate the production of proteins, which I will talk about later, but good for mentioning now. What will happen is if you decide, your cell decides halfway through the process of polyadenating, adding those A's, that you don't need this transcript anymore, it will stop adding those poly A's, and the cell's nuclear material will recognize that as not finished, and it will degrade it and break it down. you kind of have options when you produce mRNA, that you don't have to keep producing it and wasting energy if you don't need it anymore. Adding those A's, the adenines, then stops the nuclear material from breaking it down, and it also tells the nuclear pore that the end of the mRNA transcript is leaving the pore, so the pore can shut behind it. It also helps with translation. We'll look at that when we get to looking at the ribosome in the next lecture. I do want to point out the way to remember this is every single mRNA transcript starts with a lovely hat. Gee, I look good today. And this says the mRNA transcript, but the end is like, ah, as it goes on its way. When I make my transcript, it's too long. I got my cap, I got my tails, I'm looking good, I'm ready to leave, but I've got information that is not relevant to this particular protein that I want to make. I want to make the parts of my code that I need to express. I have a bunch of exons or fragments on my mRNA that comprise the actual polypeptide code, and they're interspersed with introns, intervening sequences that are not part of the code. We got to remove the introns. The introns need to stay in the nucleus. They need to stay in. The exons need to exit the nucleus. The introns, those intervening sequences, are often things like silencers or enhancers. They were helpful during transcription. They're helpful on the DNA. We used to call them junk. It turns out a lot of them are not junk. Some of them also are relevant for alternate splicing versions of this. There are sequences of your DNA that code for more than one gene, depending on how I remove exons and introns. This seems to be more common in flowering plants and in mammals than other eukaryotes. And it might actually be the reason that our DNA sequences are so much shorter than a lot of the other eukaryotes. The largest sequence of DNA on the planet, I think, belongs to some kind of moss, which is, and then close behind it, maybe an aphid, which is a little insect. Mammals and flowering plants have comparatively short sequences. Some of that is because some of our sequences overlap each other. So I could have gene 1 and gene 2, but gene 3 can be comprised of slightly different splices depending on how I remove exons. Exon shuffling does seem to play a role in doing things like the evolution of new genes. We're going to talk about that next time. We're going to also talk about this next time, but it fits here too. I break the phosphodiester bonds, I remove the intervening segment, and I reform phosphodiester bonds between the two pieces that I want to join together. I have an endonuclease that cuts the introns at both ends. Usually one of those ends, the 5' end, is marked by a guanine uracil, and at the other side it's marked by at a 3' by an adenine guanine. When I do that, I cut, choop, choop, and I glue the free OH of the side that I cut off with the new 5' side of the other side of where I cut my intron out, and ligase glues them together, forming a new phosphodiester bond. Some introns, group 1 and group 2, can self-excise themselves. That's actually coded for by the intron itself, so the intron removes itself. I also have inside the nucleus giant proteins, sometimes bigger than a ribosome, called spliceosomes. These are made up of small nuclear RNAs, snRNAs, which are put together, we say complex, but they're built together with nuclear proteins to form small nuclear ribonucleal proteins, or snRNPs. Which is, if you read that as a sentence instead of a bunch of letters, read that. Schnurps. They're called snurps. When I tried to find when I tried to find a picture of one online, because your book doesn't have any pictures, I got hundreds of these things. This is a tasty snurp. So it's super unhelpful, but it cracked me up, and I really doesn't take much to entertain me. So I was sorry. I'm sorry. Snurps. Snurps are part of the spliceosome, and this is super fascinating to me. The idea that inside of every single one of your nuclear envelopes, depending on what kind of protein that thing is supposed to make, it has a series of snurps that are appropriate only for the proteins that your cell is actually making. They bind to the 5' end of the sequence, so the donor of the splice, and terminate at the splicing acceptor sequence. And they literally fold around it and snip it based on the code in the transcript. They are specific to the protein they are snipping, which means you could be perfectly fine in your DNA code at making a gene and making a perfectly beautiful mRNA transcript with all its exons and introns. But if you do not have the genetic code for the SNRPs and the spliceosomes, you actually can't make that protein. So your gene code could look fine, but I don't know why you can't make the protein, which adds a whole other layer to genetic diseases that I don't know if we've actually had time to talk about yet. SNRPs. This is how your book drew it, which is why I was looking for images that were a little bit better. Your SNRPs are often a series of different sequences tied to proteins that are part of the spliceosome complex. They grab onto the code that they match with and they snip it. When they do the snipping, they actually fold the transcript and make a loop. As that loop forms, this causes this to break off and form a free OH. They excise the information that is not needed and leave the exon that is needed. The other exon over here is then spliced by ligase. That's really arty, I'm sorry. This other exon over here is then spliced using ligase to create our final mature mRNA with its cap, and it's all exons spliced together, and its tail at the end. This mRNA is ready for export from the nucleus. There is some editing that can happen after this. I want to highlight this here and then maybe talk about it later, depending on how the rest of the semester goes. There are some changes to the code in some cases prior to translation, post-transcription. This is, I highlighted a little bit here. It tends to be insertion deletion editing. I could not find a lot about it. Your book mentions it in passing, but it is a very, very newly understood process, but super cool if this is how we deal with in the moment problems. It talks about in an octopus and a couple other things where we have seen this post-transcriptional editing. In humans, it's known to occur in an intestinal cell protein. It's apolipoprotein B or ApoB. It does seem to be edited after the transcript itself is edited, where a cytosine is changed to a uracil, which actually changes the code to a stop codon and causes the protein transcript to really only code for the first half of it, because there's a stop codon now in the middle of it. This seems to be an adaptation to deal with infections in the intestines and is triggered when the intestine is under some infection by a parasite. I tried to grab a bunch of stuff for it, and it was really just observations of it, no understanding of the mechanisms, which means that this is something we're starting to understand, but it's likely not a singular mechanism in this one protein. This is probably something that happens under certain situations to maybe as a defensive mechanism. So kind of cool, absolutely worth looking into, but I couldn't find enough about it to make like a whole topic area I have a little bit of practice with me. I also want to highlight that my goal here is for you to build a model of transcription that's like one step more difficult than what you did in micro or in gen bio, not to add in a whole new set of processes. Your textbook ends with a bunch of, this is where we think we're going. I think there's a new edition of the textbook coming out next year. I don't have access to it. I think I almost want to wait to cover some of that, so I didn't include it in lecture, because I feel like there's a couple things in your textbook that are like, maybe it works this way. And when then I try to research it, I see we have a little bit better understanding, but not yet enough understanding for me to be explaining it to you or for me to really understand what's happening. Absolutely take a look at those last 2 segments of the chapter, but I won't hold you to any of that on an exam or in a lab situation, because it is really, really new, like new this year information. I do want you to understand things like this. The sigma subunit of bacterial RNA polymerase does what? It binds to the bacterial gene's promoter. We don't have this in eukaryotes. It binds to the region of the promoter where we are saying, this is where the gene is about to start. This is the information I want to transcribe. And it basically puts the RNA polymerase in the right position to build the transcript. I think one more question. The poly A tail of mRNA. And I'm going to go on. If you're not ready, pause me. It does all of that. It adds to the 3' end. It helps to stop the process of transcription. It is the end of it. And it's actually the last part of transcription. And it prevents the degradation of eukaryotic mRNAs in the nucleus. I have this for you. So I don't know. I was going to do a little bit of it and set you up to you if you want to stop here. But to take this into the next set of exams and to actually use it in your real life, I think it is helpful for you to start comparing and contrasting similar processes. We saw a video with elephants about PCR. We are going to do it in lab when we come back from spring break. This is the process of polymerase chain reaction. How I build DNA sequences in the lab when I'm trying to amplify them. Transcription is the process of going from DNA to mRNA. And DNA replication is the process of going from DNA to DNA, making a copy. I think it's helpful for you to make yourself a giant table, and we'll include translation down here after we did it. What happens in the start of DNA replication? What happens is I elongate what proteins are involved, literally a 1-sentence description of what happens, and then how do we stop it? And I think you should make some of your own categories to keep comparing these. And I think you all have already started to figure this out. The volume of information in biology, whether you're doing animal science or you're doing cell bio or you're pre-med, all of the volume of information, it increases exponentially. The amount of stuff that you had to learn at med school 15 years ago is like 1/5 of what a med school student needs to learn today. The amount of information is unbelievably heavy. And I can't even imagine what that is for something like veterinary science, where we're looking at multiple different species. As you do this, the best way to process this information is to start building yourself a lot of tables comparing and contrasting likes, things that are alike and things that are different. In these processes, we are all building nucleotide-based molecules. What happens in DNA replication that does happen in PCR? What happens in DNA replication that doesn't happen in PCR? And so I would put something like my initiation for DNA replication is at the origin. I use DNA helicase. I don't want to do the whole thing. I have a DNA B that grabs a hold of the enzyme, of the molecule at the origin. Things like, even if it's a small thing, a tiny description of how we actually open up the molecules, as opposed to transcription where I have RNA polymerase. That's not how you spell that. With its sigma grabbing hold of the promoter. I can't spell anything today. And then maybe when I draw it, it would look like that, as opposed to PCR, where I really just denature with heat to start the process. See if you can, over the break or as we start building a compare and contrast, where I only put the stuff that is comparing these processes with each other. I think that'll actually start to make this a little bit of a more of a manageable set of information. With that, I will see you in the next video. I'll probably see you in lecture or in lab very soon, and we'll start doing some PCR in the lab right after spring break. Building more molecules. All right. I hope you are enjoying the warmer weather, and I'll see you soon. Bye-bye.